pleasure to introduce the first speaker, Sabri. Sabri is above all a friend. He comes from NCBS. So he'll be talking about human papillomavirus integration drives oncogenic activation through rewiring of 3D chromatin contacts in cervical cancer.
Thank you, Arjit, for your kind invitation and good hospitality. Also, I would like to thank Anu and Rahul for setting the stage for my talk. Anu has explained how complex is the cancer progression, and Rahul touched upon the gene regulation and how complex it is that you have a promoter, enhancers, and then come together. How does it regulate the gene downstream of this? In this talk, I will combine both of them, especially in the context of how a foreign pathogen, which is the human papillomavirus, when it gets integrated to the host nuclear genome, how does it drive the rewiring of the gene regulation? Before that, I will just briefly explain what our lab is interested in. We are interested in cancer evolution by looking at the cancer genomes and then trying to see what are the genetic and epigenetic changes that drives the cancer progression. As we know that the cancer starts from the single normal cell, and upon some pathogenic mutations that's been accumulated in some of the genes like oncogene or tumor suppressor that provides the cells a fitness advantage so that it will propagate more, and then all the downstream clones will acquire even additional mutations that make them more fitness advantage, and then they will thrive and then disseminate to the other parts of the tissues as well in the body, and that's how the metastasis takes place. For all this evolution to take place, as with any evolution and following the Darwinian process, you need some of these events to take place, which is genetic and epigenetic one. And if we have the genome that's been obtained from here or here, we can look into the genome and ask where these genetic events and epigenetic events takes place, and can we target them. So the way we do is we are getting the data from the patient sample, again thanks to all the consortia that makes the data publicly available, and then we have a data of multiple layers, whether it can be mutations, copy number, gene expression, and so on. These are all the multifactorial data that we get from the patient tumors. And then from this, can we look into some of the processes like somatic mutation, like how the mutations happen in the genome in the first place, and then which mutations are functionally relevant, which we call them as the driver mutations, and that shows the signals of positive selection, because this cancer follows the Darwinian evolution, so you would expect to see positive selection if you look at the gene which are mutated across multiple samples. And then we're also interested to look at the tumor immune interaction, because when the tumor starts and grow, you always have a constant check from the immune cells, because immune cells are good at finding these faulty cells and then kill them. But tumor somehow evades from these immune destructions so that it could thrive in the context of the immune pressure. So what we want to do ultimately is, once we learn all these things, can we assign what are the key genetic or driver alterations that are present in each individual, and can we use that information to predict the prognosis of how likely the patient would respond to a treatment and for how long. So this is the ultimate that we want to get for finding the biomarkers. So today's talk, I will be focusing on the HPV as a driver. This work was primarily done by a graduate student, Anurag. And the HPV is been associated with cervical cancer for a very long time. So the way it works is that, let's say that this is our cervical epithelium. When the HPV virus get into that, most of the time, our immune system will detect them and clear it off. So 95% of the time, our immune system is good at detecting this virus, so as with any other pathogen coming to our body, and clear it off. But in any unfortunate situation, if this virus gets affected the basal cell, which is the very lower bottom of this epithelial stratum, and this basal cell has a property like a stem cells. So they can divide and then form all this upper layer whenever this upper layer cells would undergo cell death or something. Because of this stem cell-like property in the basal, it divides every time, and virus take advantage of this replication process that is present in the host and multiply its own copy of the virus as well. So this is how the basal cell will go and produce more of the upper epithelial cells, and so the virus go replicate along with that. When you have more copy of the virus, of course, it can affect the neighboring cells. And over a time, so at this phase, this is only a group of fast dividing cells. It's still not a problem at this stage if people go for the regular screening, that's called something called pap smear. When they detect at this stage there is some abnormal growth, and if there is a presence of HPV, people can do some treatment at this stage and can get cured from this one. So that's how the testings are important. So this will help to prevent the disease to further grow. If we miss the screening part, and the virus may get entered to the host genome as well, and then it gets integrated as well. So this is a DNA virus. So this is how it looks like. It is around 5 to 7 kb. It has this important gene called E6 and E7. These are all oncogene that has been well studied in the last 3 decades. And then you also have this E2 gene, which function as a regulator that controls the expression of the E6 and E7. When this virus exists as this episome, like this ring-like structure, the expression of the E6 and E7 is well controlled by the E2, and it's not that problematic at this stage. But when it gets integrated to the host, this happens because if you have a break here, like a DNA break, this becomes linear, and then our host genome is also linear. And then if there is any repair that happens between these two broken DNA, like non-homologous end joining, you can get this entire virus gets integrated to the host genome. Okay? This is how the virus now gets integrated to the host DNA. And most often, the break happens at the E2 stage, E2 gene, and because of the loss of the E2, you don't have a regulation of this E6 and E7 as it used to be in the circular form. So the aberrant expression of the E6 and E7 downregulates some of the P53 and the RB pathway and drives these cells much more invasive tumor than it used to be in the early stages. Okay, that's why it's important to know if the integration is present in the tumor, then it can drive these cells even crazy. So a lot has been done, studied in the context of E6 and E7. But if you think broader in terms of the gene regulation, as Rahul was alluding yesterday, that our genome is being organized into topologically associating domains. The regions within the topologically associated domains, or TADs so-called, have a lot of gene regulations within them. So the question that we asked is, like, if this integration happens in a particular TAD, how does it affect the regulation of the host gene? Can that also drives or favors the cancer progression? People have looked at it, where the integration happens and whether does it affect the expression of the host gene as well. People found that integration closer to the MYC can activate the expression of the MYC gene, and also some of the recurrent integration in the open chromatin region also upregulates some of the host gene nearby. But all these cases, people have looked at only the close integration, like the integration is at the promoter region or within the genic region. So what we wanted to ask is, like, whether the integration that are further away in the intergenic region can also affect the expression of some of these host genes. And we do this at the multiple layer. So we have this host chromosome like this, and you can have the heterochromatin region like this, where much of the regions are inactive. And you can have these active regions where there is a lot of transcription takes place. And then you can also have these long-range chromatin interactions that forms the enhancer promoter loops like this. So we asked how the integrations could affect this configurations, and thereby drives the expression of some of the host genes. For this, we have taken the HPV integrations from cervical patient samples. We got around 617 from around 200 samples, mostly from TCGA and other studies. And the way people find integration is we have the DNA sequence from the tumors. Most of them are short reads. So if you have a DNA like this, you would get reads from the non-integrated region and also from the integrated regions. And then by doing the split read mapping, you would find the reads that partially maps to the HPV genome and also maps to the host chromatin. And by taking advantage of this split read, meaning that part of the reads is mapping to the host and the part of the read mapping to the virus genome, we can find where the integration takes place. So this is how we have identified the 617 integrations. And then to check whether this integration affects the host, we are using this chromatin information from the cell lines, because unfortunately we don't have this chromatin information from the respective tumor cells. So we are using the closest cell line, the HeLa. Many of you are aware of this cell line. This is derived from the cervical cancer. Sorry. And we also used normal epithelial cells as a control as well. So first thing we asked is like how the integrations are distributed across this chromosome when you take this active versus the inactive chromatin information. So the active means that these are all the regions that are enriched for chromatin marks that supports the gene regulation, active gene regulations, whereas the repressed regions are kind of region that is tightly packed and there is no active transcription takes place in that region. When we looked at the distribution of the HPV integration in these two broad segmentation of the genome, so the dark blue is the observed integrations, and then the light one is the expected. So the expected were derived based on the sequence context, because as I mentioned, the integration happens through the non-homologous end joining, so meaning that there is a small overlap between the HPV DNA as well as the host DNA. By looking at that similarity, we can expect we can ask where do we expect more of this integration in the genome. And when we look at these numbers, you can see that in the inactive chromatin, we expect more integrations because these regions are enriched with the repeats. And so it's an ideal place for the integration to happen. And we observed the integrations are closer to the expected. Whereas in the active region, we expect less integrations, but what we observed is slightly higher integration as compared to the inactive region, suggesting that the integrations that are in the active region perhaps get selected in the tumor progression, and that's why we see more integration in the active regions as compared to the inactive regions, especially taking into account the expected count. Then we asked, how does the integration affects the expression of the nearby genes? So for this, we have considered all the genes that are present in the TAD. So as Rahul mentioned yesterday, that you have this Hi-C map. And from this Hi-C map, you can derive the topologically associating domains. And these are all the domains where much of the gene and regulatory regions interact more often than the regions outside. So we take all the genes that are within the TAD and ask how the expression of the genes in the TAD look if we have an integration. So here we are looking at the expression of the nearby genes in the TAD that have integration, and also we looked at how the expressions are in the neighboring TADs as well to see whether the integration affects across the chromosome or it's very local. So what you are seeing here is the expression level. It's on the y-axis. Each dot is a sample, and the one in the orange shows the expression of the nearby genes in the TAD that have an integration. And then we are comparing that to the expression coming from the samples that don't have integration in that particular TAD. So by looking at how the integration affects the expression in the TAD, we can see that TADs that have integration have slightly higher expression as compared to the samples that don't have integration, suggesting that integration can affect the expression within the TAD. So if you go beyond the TAD with integration, how does it look? Unfortunately, we don't see any change in the expression in the nearby TAD, suggesting that whatever the changes that's happening is more constrained to the TAD that have integration, and the neighboring TADs are unaffected, which is good because the host chromatin is somehow preventing the influence of the HPV integration to the regions that are outside of the TAD. Or somehow the insulator property that are at the TAD boundary is preventing the HPV integrations to interact with the regions outside of the TAD. So within this, you can have TADs with one integration or multiple integration. So we asked, how does the different number of integration correlate with the expression? As we can see here clearly that if the TAD has more integration, obviously the expression level change or expression change is much higher than the other TADs which have lesser number of integrations. Okay, this again confirms that the integrations can affect the expression of the nearby host genes. Given that there is an expression change, we asked if there is any oncogenes by chance or any tumor suppressor genes by chance in these TADs with the expression change. So here what you are seeing is a heat map. The x-axis is the different sample, and y-axis shows the different TADs. And here the darker blue indicates that there is an integration in that particular TAD from the respective sample. And we can see here a nice mutual exclusivity, meaning that not on any sample have more than one integration in a particular TAD. So it's kind of nicely segregated, suggesting that the integrations are very precise. But in this blue TAD, you may have one or multiple integrations, but it's not spanning across multiple TADs. And if you look at what kind of oncogenes are tumor suppressor in this one, we could find that the TAD that have many integration across samples have MYC and PVD1, followed by TP63 and then ERBB2 and others. Okay. We asked whether the expression of this oncogene is also being affected. So in the interest of time, I focus on the MYC and PVD1. We could see indeed TADs with integration have higher expression of the MYC as compared to the samples that don't have the integrations. Then we asked how far the integrations are from the genic region. So here on the x-axis 0 corresponds to the MYC genic region. And then each dot here is the integration from the respective samples. And then the distance tells us that how far the integrations as compared to the gene start region. And we could see that many of the samples have integration at or very close to the MYC region. And there are some integrations that are far away. Okay. So we asked how the integrations here could affect the expression. We thought that perhaps in the integration that are closer to the gene, it can affect the expression by doing the transcriptional run through, meaning you have this viral region getting integrated, and this have some LCR region, which are local control region, having some promoter-like function. And because of this strong promoter, now the gene that are downstream of that can also drives the expression even more. Or whatever the active transcription happening, all the downstream gene also gets carried away by that. In case of integration that are further away, for example, in this case, which is roughly around 500 kb, we thought that perhaps this long distance could still regulate the expression of the gene through this chromatin looping, like two regions that are far away but can come close together and regulate the expression of this one with the help of CTCF and cohesin. To test that, we have taken the Hi-C data from the HeLa cell line. HeLa cells also have HPV18 integrations, that is 500 kb away from the MYC region. So it was an ideal candidate for us to check. And then we looked at how the chromatin interactions are between the HPV DNA versus the host DNA. And we could see that much of the interaction happening within the chromosome 8. This is where the integration has happened. There is a lot of chromatin contacts between the HPV18 and the chromosome 8. Then we went to check which genomic region exactly having much of this chromatin contact, and we could see in the TAD that have the HPV integrations. Then we asked how the interaction strength between the two different chromatin regions in the TAD with integrations as compared to the outside region. We could see much of the chromatin interaction between the HPV18 and the TAD with TAD 3189 is happening mostly within the TAD, and less number of interaction is going away from the TAD, meaning that if a TAD have this integration, all the chromatin interactions are within the TAD, and the interaction that is going away is very less. Okay, I'm almost done. Okay. Then we asked within the TAD which region have a stronger interactions, and we could find, as expected, MYC and PVD1 has a very strong interaction as compared to the other regions within the TAD. So we checked, we performed 4C analysis to confirm whether there is any chromatin interaction between the integrated HPV and the MYC promoter. And this was done in collaboration with the Dimple Notani at NCBS. What we have done is we have designed primers at the integrated site and then asked what other regions are interacting with the HPV18. And as we have seen in the Hi-C data, we could see much of the interactions are happening at the MYC and the PVT1 promoter regions. If there is a chromatin interaction, of course, we would expect the gene expression to be different as well. In HeLa, we know that all the chromosomes have the 2 copies, one inherited from the parent and one inherited from the mother. In HeLa, the integration happened in only one chromosome of one copy of, or one allele of chromosome 8. The other allele is still intact. So we asked how the expression coming from one allele or the HPV integration drives expression mainly from the allele where it's integrated as compared to the other allele. So we have done this allele-specific expression by taking the SNP position as a reference point, and we could see much of the expression from the MYC and PVT1 comes from the allele where the integration has happened as compared to the other allele. Again, confirming that there is a chromatin interactions, and then it drives the expression of this one. So we hypothesized that this long-range chromatin interaction could be driven by the CTCF and cohesin. Can we test this? So we looked for the data set whether the CTCF or cohesin is knocked down, and then they performed HiC on it. So we were fortunate to got one data set where they have conditionally knocked down CTCF and cohesin, and then they performed HiC on it. And then we looked for the chromatin interactions between the HPV-18 and the TAD where it was integrated. And we could see that in SCC1 knockout, which is one of the cohesin complex, the chromatin interaction has drastically down. Whereas with CTCF knockdown, there is no change in the chromatin interactions, suggesting that cohesin is the major player that drives the interaction between the HPV-18 and the MYC promoter region. So whether the reduce in the chromatin interaction will also influence the gene expression, and as we expect, the MYC expression has been significantly downregulated as compared to the control samples which don't have the CTCS or cohesin knockdown, suggesting that the cohesin is involved in the looping between the HPV-18 integration and the MYC promoter. So based on this, we proposed this model that in the initial stage of the infection, the virus exists as an episome. It's not that lethal at this stage, but over a time when it gets integrated, not only it drives the expression of the E6 and E7 from the viral region, but it can also act like an enhancer that drives the expression of the oncogene within the TAD that has an integration. So this suggests that the integration acts like a multi-hit driver, meaning there is some oncogenic event coming from the viral DNA, and also this entire event can also influence the expression of the host gene as well. So in the screening process, we are trying to propose that if there is an integration being detected at the early stage, it's better to follow them up at very closest stage so that we can avoid the formation of the invasive tumors. So we are trying to collaborate with Dr. Rama at Kasturibai Medical College, where we are trying to get access to these patient samples, especially at the early stage, to check how often we see this integration, and if the integration is any of these stats that we have seen, can we follow them up very close to see how likely that drives into the invasive tumors. So this is work in progress. Hopefully next time I will present some of these results from this study. With this, I would like to thank my group, especially Anurag, who has done majority of this work, Dimple, Johnny, and Rama for all the collaboration, NCBS and India Alliance for funding, ICGC and TCGA for making the data publicly available, and thank you for your time.
Thank you, Sabri. Wonderful talk, and thank you for being well inside the time. So we have 5 minutes for Q&A. So, yeah.
Thank you so much, sir. That was a great talk. So I hear that you are saying that viral integration is happening in only one copy of the allele. I mean, chromosome 8. Then, I just curious to know, is that integration is biased towards a paternal or maternal chromosome? I mean, have you observed anything like that?
Yeah, good question. We haven't looked into which allele is that, because right now we can, unless if we have a trio information, meaning that if we have a genome from the parent, from the father and mother, we can find which allele is the parental or maternal, and then we can find the segregation. Right now we could not say that, but just by looking at the SNP frequency, we can say which of the allele have the integration. Yeah. But we think that there is no influence of which allele it is. It happened in one of them because if you look at the cell, even though they are autosome, the chromosome positions are quite different. So they don't occur very closely, they are far away.
You showed some data where you showed that the integration has some specificity in terms of the specific oncogenes which it targets.
Yeah.
And at the same time, you showed that there are long-range interactions where the integrated part itself acts like an enhancer.
Yeah.
So my question is, are there any specificities of this enhancer kind of activity which is related to only certain kind of TADs, or this is a much more generic feature which, if integrated in some other TAD with some oncogene in a synthetic setting, would also lead to a general upregulation?
Yeah, very good question. We think it's a very generic process. So as with any of the plasmid construct, that if you bring the viral promoter, the expression of whatever the gene that we are tagging will go really higher. So it's the same principle we are applying here, that this viral integration, as long as this LCR region within that, which is actually the promoter-like sequence, as long as that is in the intact position, it could act like an enhancer or promoter. Yeah. So we need to look at how the integrated sequence is intact or not. Yeah.
Hey, great talk. Do you think it can be predicted where this integration will happen? Like by using machine learning or something like that, and would that help? And making some downstream predictions on top of that as well.
Yeah.
In terms of.
Yeah, it's very interesting. So right now, the way that we compute expected or based on this sequence composition, because it undergoes this non-homologous enjoining, it needs a little bit of similarity between like sequence pairing between the host and the viral. So that's how the non-homologous enjoining work. I think if people will do this kind of match between the two DNA, I think we can predict the integrations. Yeah.
Questions?
Yeah, just curious. So I believe HPV causes only a small number of cancers, cervical and some other reproductive tract and throat, I think. And does it integrate? Is it known as? Does it integrate into the genome in those other tissues? And why doesn't it affect other tissues?
Yeah, very, very good question. In a patient perspective, it's fortunate that it's not affecting the other tissues. But in terms of the tumorigenesis, I think this is something related to the mucosal cell type and so on. But they don't see much often. But people have some skin lesions which are related to the HPV. Yeah, but this specific strain is related to. No. Yeah.
Okay. Any further questions? If not, then let's thank Sabri for this wonderful talk again.
Yeah. Thank you.